Hi, welcome to new data science project clustering virus nucleotides using k-means. In this project, we will apply k-means clustering to group virus nucleotide data and visualize the clustering process. We'll start by introducing clustering, which is a method of grouping similar data points together without labels. This technique is widely used in areas like market segmentation, social network analysis, medical imaging, and anomaly detection. K-means is one of the most popular clustering algorithms. Next, we'll dive into how k-means works. Initially, we start with a dataset of scattered data points and place random centroids. These centroids will guide the clustering process. The first step is to input the data and initialize centroids. After this, the data points group around these centroids, forming clusters. The next step involves calculating the distance between each data point and its centroid. This helps assign each point to a specific cluster. Once the initial clustering is done, we adjust the centroids to better fit the data points. This adjustment process helps to refine the clusters. After several iterations, the centroids stabilize and the clusters are final. Now, while working with data, sometimes it's too high dimensional to process effectively. This is where dimensionality reduction comes in. It simplifies the data by reducing the number of dimensions while keeping essential information. To reduce dimensionality, we use PCA, Principal Component Analysis, a technique that identifies the most important features in the data and projects it into a lower dimensional space. By sorting and selecting the most critical features, PCA helps in simplifying the dataset for better processing. Another technique we use for dimensionality reduction is t-SNE, T-Distributed Stochastic Neighbor Embedding, which visualizes high-dimensional data in a 2D plane, grouping similar data points close to each other, making it easier to interpret. For our project, the dataset contains virus nucleotide sequences. These sequences will be pre-processed and then clustered using k-means. To speed up the clustering process and make it more efficient, we integrate PCA to reduce the dataset's dimensionality before passing it into k-means. After clustering, we visualize the results using t-SNE, which will help us see how well k-means has grouped the data. By following these steps, PCA, k-means clustering, and t-SNE, we are able to cluster virus nucleotide data effectively. Thank you for watching.